The Earth is one big system, and the ocean is an important part of that system.
My name's Emmett Duffy, and I'm the director of the Smithsonian's Marine Geo program.
We're here in Wachapreague, Virginia for a Marine Geo Bio Blitz. This year we're focused on small invertebrates.
I've done 3 Bio Blitzes.
1 Bio Blitz.
10 Bio Blitzes.
1 Bio Blitz.
2 Bio Blitzes.
Maybe 25 Bio Blitzes. This is in a part of the world where you think we know everything. And any Bio Blitz will show you that the planet and especially the oceans are incredibly poorly known despite everybody thinks we know this stuff. And if we don't know what we have, how can we manage the ecosystems? How can we understand change? How can we plan for the future?
Although the Chesapeake Bay has been studied extensively for a long period of time, there hasn't been a lot of collections recently in the last several decades to century. We hope to fill in some of these gaps.
The Bio Blitz is important because trying to quantify the taxonomy with the barcoding DNA, I think it's going to be real helpful down the line for biodiversity studies.
We know that the world is changing, climate change is happening, and this is a great way to get a baseline of the species that are present here and other places, and that is giving us a baseline for long-term data so we can kind of see how species change over time.
I'm Matt OGBURN. I'm a research ecologist at the Smithsonian Environmental Research Center and coordinator of the Chesapeake Bay site for the Marine Geo Network. The first step of our Bio Blitz is to collect samples in the field. We've been collecting the samples using a variety of different methods, like pulling a trawl behind a boat or pulling a dredge which rakes through the sediment. We've also been coring the sediment for worms and clams and using a variety of other field methods to just collect animals and bring them back to the lab. Then we sort the sediment or grasses or other things that we collected. We do the sorting to pick individual animals out that we're interested in identifying. Once we've identified as best we can a particular species, we collect what's called a DNA barcode analysis, which is just a genetic analysis that gets a standard short segment of DNA that helps us identify that species. If someone were to collect it again, they wouldn't have to do the hard work we're doing here to actually try and identify it based on how it looks. We take a photograph of each of the specimens so that we have a photographic record of it that we can keep in digital collections. Finally, we preserve each specimen so that there's a physical record of it that will be held in the Smithsonian National Museum of Natural History.
We still depend on the ocean, need to understand how it works.
The basic understanding of the oceans is really limited, and these kinds of surveys help us move forward rapidly.